Sorry for the slightly long title. Hopefully you can all hear me well. So this talks about a package I've been working on for where you have a matrix of some sort of observation and an associated matrix of precision weights. So if you've used voom, voom is an example of that. I basically wanted something a fair bit more general than voom, but with the same general idea. And this uses these modern data types like summarized experiment. It can use delayed array. Most of the functions are parallelized. So first off, what do I mean by weights? So there's a few different senses of weights. I'm using it in the sense of, same sense that's used in weighted least squares by functions like lm or lmfit. So we're minimizing the sum of weighted squared residuals by choosing coefficients. And that's how you fit a linear model. And then the optimal weights in this case are one on the variance of noise in the observations. So that's optimal in the sense of being a blue estimate. And if your noise happens to be Gaussian, it'll be a maximum likelihood estimate, though that's not an assumption we need to make. Another advantage of using weights in this way is that we'll then be able to work out the variance of error in the estimates that we're making, and then we can do valid statistical tests. And then finally, a weight of 0 we can use to indicate missing data. So this package will deal with missing data quite well. A particular case of weights that's quite important is where your observations are actually averages of some underlying observations and varying numbers of underlying observations. In that case, appropriate weights are the number of underlying observations that you're averaging. Okay, so example of this. So this is a PAT-Seq dataset. PAT-Seq is an RNA-Seq method that sequences 3' ends of transcripts, and somewhat unusually, it sequences into the poly-A tail. So we get some information about the poly-A tail length from seeing a poly-A sequence in the reads. It's only an indication. Like the sequencer struggles with it a bit, but it does give us information. So what I'm going to do is for each gene in each sample, for all the reads we have that have this poly-A sequence, I'm going to average the observed tail length. And then the weight is going to be the number of reads that we see. So this particular example is yeast cells that are in synchronized cell cycling. And there's 2 strains of them, and we see 2 cycles. And first up, I'm going to fit linear models to each gene in this data set. So now, after fitting a linear model to each gene, this is now plotting all of the residuals from all of the genes. So we've got residuals on the y-axis, and then I've put log read count on the x-axis. You can see as you get more reads, the noise goes down. Now these lines, the center one is the mean trend, and then these outer ones are plus or minus 1 standard deviation. So you can see like that having no weights at all isn't really a good fit to this data. Now, if we're using weights, the appropriate way to plot them in this sort of plot is we'll multiply the residuals by the square root of the weights. And if that's a good set of weights, then we'll get straight lines. We'll get a uniform value for the standard deviation. But you can see in this case, well, it's fixed up the left-hand side here, but then the right-hand side is, we've sort of stretched that out too much. So there's some biological variation in this data that a somewhat naive choice of weights hasn't accounted for. So can we do about that? So we've got an initial set of residuals. I'm going to put them all into a big long form table. And what I'm going to do is make a model of the squared residuals based on various things. And then from that model, using one on the predictions from that model as the new set of weights. So this is essentially modeling the noise in the data. This model, we can specify it as a linear model. So what I'm going to put in this linear model is a spline curve through a sort of initial guess at the weights, which were the read counts, and I may as well put in a spline curve through the mean tail length as well, so we can get a mean variance trend. So this is fairly flexible. Okay. So now this is the calibrated weights from that function, and you can see now these weighted residuals, the standard deviation is uniform relative to the read count. So that's what we want for good fits and also for statistical testing. We can plot against other things, and so this is against mean tail length, and again we have a nice uniform standard deviation. Okay. So a few comments on this. This is similar to the voom method, but somewhat more general because we can throw in other pieces of information rather than just the mean. It's actually similar to something called a double GLM, to like one iteration of a double GLM, and similar to a double GLM specified using these two linear models, the normal design linear model and then this linear model for the variance. And that linear model for the variance gives you quite a lot of flexibility. You could put in like factors to do with particular groups of columns or particular rows or whatever you think of. Okay, so what can we do with this? So the most immediate thing is differential testing. So these weights are compatible with limma. One thing here, we're using weights fairly aggressively. So some genes may have quite a lot higher weight than others. And that'll mean even with small differences, they'll get very significant results. So we maybe don't want to be sorting by a p-value. Treats is a principled approach to getting around this problem. It lets you test relative to a threshold rather than relative to a null hypothesis of exactly zero. And then I've got a package called topconfex, which will essentially do the treat test for a bunch of different thresholds and then sort genes by the largest threshold that they're significant at. So that's essentially a confidence bound on the difference we're looking for with our multiple testing correction. Okay, so here's an example of this in action with this package. It's got a nice, it's basically got its own implementation of this technique. And so with this data set, it's a bit awkward to come up with differential tests because there's no replicates, but we can look for difference between the strains. We have like difference between the strains here and then a confidence bound on that, and the results are sorted by that. We get a bunch of interesting looking results. One thing, once you do have these calibrated weights, you can start plotting standard deviations on these individual measurements, which is nice. But yeah, we're limited by this design without replicates. So what are some other things we can do once we have nice precision weights for a data set? One thing is we know how much noise we expect, so we can look for noise in excess of that. So I came up with a test relative to a threshold based on this idea, plugged that into topconfex. And then this is looking for noise in excess of a very simple model, a simpler model than we used before, where we're just assuming all the tail lengths are equal. And so these are the sorts of genes we're finding. And these results are, again, they're given in units of standard deviations of tail lengths. And one thing you can notice in these last couple of genes that's found down here, they're actually showing a bit of cell cycling, which is what this particular experiment was about. So this is similar to finding marker genes in single cell. So without much in the way of it, a model. A thing we can do is look for components of variation in this. So essentially the weighted version of PCA. Again, I've got a function for that in this package. I've asked it for 3 components. It automatically varimax rotates these, so they should be interpretable, fingers crossed. In fact, they do look fairly interpretable. And with 3 components, the 3rd component is actually this cell cycling component, it looks like. And so we can get a list of genes associated with that. So that's 3 different ways you can get useful results out of a data set once you have these calibrated weights. So now, this package, like what I want to do with it is I want it to be a fairly generic sort of thing that, like, there's many sorts of data where you get a matrix of measurements of some sort. So to quickly demonstrate it with another data type, alternative polyadenylation, and then this is a 10x data set, single cell, and spermatogenesis. And we get various genes where there's a switch to a shorter form of the gene. There's 10x is a 3' focused sequencing method, so we get peaks associated with different APA sites. So there's starting to be a few pipelines that will call peaks and quantify them and that sort of thing, but then, like, how do we pick out interesting results from that? So I came up with a measurement that I think gets at what I'm interested in here. I wanted it to be signed because we have, like, general tendencies to the shortening or lengthening. So essentially, for each gene, give each site a score indicating upstrandness or downstrandness, and then each UMI belongs to a particular site, and then for each gene in each cell, calculating the average of the scores of each UMI. But anyway, I came up with something, it measures something I'm interested in. Same workflow, you do the calibration step, just provide to this calibration step information we have. So again, we have a read count, or well, a UMI count. We also have some idea of, like, how variable these will be based on their distribution. So these plots are very small, but basically the message here is do a lot of this style of plot to make sure it's all working. And then those 3 different ways of interrogating the results we could apply here. So here, this is a differential test between two stages in this sperm development. And the thing I wanted to point out here is this is working even with a lot of missing data. And, like, the top gene by this top convex method actually has a lot of missing data, but we're still confident it's a large effect size. Okay, so that's the package, hoping it's going to be fairly broadly useful. So I'd like to thank Further Buyers, who I've worked with for a few years now, who came up with the PAT-Seq technique. Bernard Dichtel was involved in making that particular data set. Sarah Williams has been writing the polya pipe package, and David Powell as well has been involved in that. So, thank you.
Thank you very much, Dr. Harrison, for a great talk. And any questions, please type in the chat box. And you can look in the chat box to see if there are questions for you. Let's see. If not, I'll go ahead with one and just to see other people while waiting for other people. For this, okay, so have you did like some evaluation between your basically it's weighted by the variance, right? Like opposite, like the reciprocal of variance. And the more variance, you know, the less weight you're going to give. And from one, how you compare to the method for dispersion, like all these different methods for HR or limma or I think the D-Seq too. Have you done anything like to compare your method to the others? Have a benchmark data for, you know, bulk RNA-Seq and even
yeah, that definitely sounds like a good idea. So, like, I have done a tiny bit of comparing, like, the results to voom's selection of weights instead of in terms of those weighted residuals. But yeah, I haven't really done a lot of comparison. My main focus has been like applying it to these weird data types that I don't have a method to compare it to.
Okay, thank you.
Yeah, that's a good idea.